Hello everyone, I am Aruna Punna, working as junior lecturer in botany in Government Junior College for Girls, Bongir. Today our topic is plant growth and development. In this class, I am going to teach you about physiological effects of gibberellins and cytokinins. The physiological effects of gibberellins. Up to now, more than 100 gibberellins reported from different fungi and higher plants. All these gibberellins denoted as GA1, GA2, GA3 and so on. First time in Japan country, pad seedlings affected by Bakane disease, which is caused by Gibberella fusicori. Gibberella fusicori, it is one kind of fungus. From this, GA3 is first isolated by the scientists. So GA3 is the first discovered one and it is studied intensively. By spraying gibberellins, we can increase the length of grape stalks. What is the use of it? Here you can see these two grape bunches. The difference is this grape bunch is very short and in this grapes are compactly arranged. But here the grape bunch is very lengthy and the grapes are freely arranged. In this you can observe here grape stalks are very lengthy. What happens if the grape stalks are very lengthy? In this diagram, grape stalks are very short like this. So here what happens if the grape stalk is very short, only some grapefruits only grow well. The remaining fruits, they won't have sufficient space to grow. That is why the remaining fruits are very smaller in size. But by increasing grape length, what happens? Each and every grapefruit gets sufficient space and grow well. That is why by spraying gibberellins, the grape bunch length increases and each and every fruit grow well. In this way, the grape yield increases. And by spraying gibberellins, apple fruit size also increases. In which way? Here this apple fruit is very small in size. This is normally grown. That means not sprayed by gibberellins. But here this fruit is very large in size. Fruits is elongated. According to the length, the diameter of fruit also increases. Overall fruit size increased. By spraying gibberellins only, this fruit size increases happens. This type of fruits get more price in the market. By spraying gibberellins, we can delay the senescence. Senescence means ripening fruits. For example, you can take tomato cultivation. In tomato cultivation, the main problem is after harvesting, it is very difficult to store tomatoes for a long time. Whenever the tomato production increases, automatically in the market, tomato price decreases. You know all, sometimes 1 kg tomato cost maybe 100 rupees and sometimes maybe even one single rupee. So this situation leads to great loss to the farmer. How can we stop this situation? By spraying gibberellins on tomato crop, what happens? Delay senescence means ripening of tomatoes stop. So fruits can be left on the tree for a long time and extend the market period. Here, this tomato crop is sprayed by gibberellins. That is why tomatoes without ripening, they can stay for a long time on the plants. That is why there no need to harvest complete crop at a time. Farmer can harvest the tomatoes step by step in small quantities and immediately shift to the market. So what happens in the market? There is no overproduction and there is no decrease in the price. So always in the market, the tomato cost is balanced. That means 1 kg tomato approximately between 20 to 40 rupees. In this way, by spraying gibberellins, we can stop the loss to the farmer. And these gibberellins speed up the malting process in brewing industry. Usually brewing industries produce alcohols like beer, whiskey, wines like products. By malting process. In malting process, what happens? The cereals like paddy, wheat and corn seeds should be taken. These seeds soaked in the water. So the seeds germinated and on the germinated seeds, hot vapor must be exposed. Then what happens? By hot vapor, the proteins and carbohydrates which are present in the germinated seeds broken down and some enzymes also become activated. That is called malting. Usually for the malting process, approximately 4 to 6 days time will be taken. But when we add these gibberellins in the malting process speed up. That means they won't take 4 to 6 days. Just within 2 to 3 days only, malting process completed. That is a useful aspect of gibberellins in brewing industries. And by spraying gibberellins, we can increase sugarcane yield. In which way? So here these are the sugar canes. Here two diagrams are there. In this diagram, you can see the internode region is very short. This is nodal region. This is also nodal region. Nodal region means wherever leaf grows, that is nodal region. Between two nodal regions called internodal region. Here the internodes are very short. So this is normal crop. This crop is not sprayed by gibberellins. But here, see this diagram. Here the internodes are very lengthy. By spraying gibberellins, internodes become lengthy. Automatically stem length also increases. When the stem length increases, sugarcane production also increases. By spraying gibberellins, we can increase sugarcane yield approximately 20 tons per acre. And by spraying gibberellins on juvenile coniferous. Juvenile means young coniferous. What about coniferous? Here, these are the coniferous trees. Coniferous trees are always in conical shape. That is why this name coniferous has been given. These coniferous trees belongs to gymnosperms. Gymnosperms always produce directly seeds only. They never produce fruits. Here, these are the cones which are developed on the coniferous trees, and these cones produce edible seeds. Here, what happens? Generally, after completing 3 years of age only, coniferous trees start to produce these cones. But here, by spraying gibberellins on young coniferous, just after completing 1 year age only, the plants start to produce the edible seeds. That means cones. And at the same time, one more interesting thing, just for the seed development only, plants take nearly 1 to 2 years of time. But by spraying gibberellins, within 1 year only, seed development completed. In this way, by spraying gibberellins on juvenile, means young coniferous, we can fasten the maturity period of the plant, and that situation leads to early seed production. Gibberellins promote bolting. Bolting means internode elongation just before flowering. The bolting is most common in beetroot, cabbage, and many plants with rosette habit. Rosette habit means in the rose flower, you can see the circular compact arrangement of many large petals. This arrangement is most common in rose flower. That is why to this arrangement, rosette habit name has been given. This rosette habit is common in beetroot, cabbage, and so many plants. In cabbage, you can see clearly the rosette habit. This is cabbage plant. This is apical bud. Apical bud is used as edible part. So here you can see the longitudinal section of cabbage. This is stem apex. Around this stem apex, so many young leaves are compactly arranged and overlap by one another. Due to compact arrangement, you can see the internode region between two nodal regions. The region is called internodal region. So this internode is very short here due to compact arrangement of leaves. But what happens if we won't cut this cabbage and let it on the plant? What happens after few weeks? The cabbage plant produce inflorescence. Just before flowering, before producing inflorescence, the internodes elongate by the activity of gibberellins. That is why finally the inflorescence become very lengthy. In the inflorescence, you can see clearly the elongated internodes. This is by the activity of gibberellins. And physiological effects of cytokinins. Cytokinins have specific effect on cytokinesis. What is cytokinesis? This occur in cell division. So here this is one single cell finally divided into two cells. In first step, the nucleus divided into two, that is called karyokinesis. Here, nucleus only divided, but not cytoplasm. Around the nuclei, cytoplasm is present. So, by wall formation, cytoplasm also divided. This wall formation is called cytokinesis. So, this cytokinesis activity takes place by the cytokinins. After cytokinesis only, two cells are completely divided. And first time, the cytokinins discovered in the form of kinetin. Kinetin is a modified form of adenine. Adenine is nitrogen base. It is kind of purine, which is present commonly in DNA. So this kinetin isolated from autoclaved herring sperm DNA. Herring is one kind of marine fish. It is name of the fish. From this herring marine fish, sperm is separated. In the sperm DNA is present. This sperm is autoclave. Autoclave means it is one kind of electric pressure cooker. In this proper temperature and pressure is maintained. So in this, the herring marine fish DNA is sterilized. So in the sterilization, what happens? Any microorganisms are present in that, they will killed. So in this way, by the process of autoclaving the herring sperm DNA, kinetin is discovered. But kinetins are not present naturally in plants. In plants, what is naturally present means cytokinins are present in the form of gelatin. This gelatin isolated from corn kernels and coconut milk. The gelatin also have same cytokinin like activities. After discovery of gelatin, so many natural and synthetic cytokinins are identified, and these natural cytokinins synthesized in the plant body wherever rapid cell division occur. Here, usually in the plant body, in the root apices and developing shoot buds and young fruits, cytokinins are produced. This is plant body. The below one is the root system. The above one is shoot system. This is root apex. Here, repeated division takes place by the activity of cytokinins. That is why root system grow very lengthy into the soil. And this is shoot system. This is shoot apex. Here also, due to the activity of cytokinins, repeated division takes place and stem grows very lengthy. And these are the axillary buds. Axillary region means the upper angle which is present between leaf and axis. So in the axillary regions, axillary buds develop. These axillary buds, due to the activity of cytokinins, developed into lateral branches. So in this way, by producing so many lateral branches, plants become bushy, and in the young fruits also cytokinins produced. So due to the repeated division of by the activity of cytokinins, young fruits become larger in size, and cytokinins produced even in the young leaves, because usually young leaves are smaller in size. So by the activity of cytokinins, the young leaves become larger in size, and at the same time cytokinins increase the chloroplast division. That is why when the young leaves grow into larger size, automatically chloroplast division also increases, and cytokinins produce adventitious shoot formation. What is adventitious shoot formation? Here, adventitious shoots develop from leaf, stem, or from even root also. Here, this is bryophyllum leaf. Along edge of the bryophyllum leaf, you can see adventitious buds developed. Each and every adventitious bud developed into a young plantlet, and here directly the trunk of tree is producing some branches. These are called adventitious buds, and in Karvepaku plant also the root system produce so many buds. Each and every bud, adventitious bud, developed into a plantlet. That is why you can see around the Karvepaku plant so many young Karvepaku plants are there by the activity of the adventitious buds which are producing from the root system. Here, these adventitious branches produced from anywhere, from leaf, from stem, or from root, but except axillary buds. You know, axillary bud means the upper angle which is present between leaf and stem. So, except axillary region, adventitious roots develop from anywhere, from leaf, from stem, from root system. Axillary buds developed and produce only lateral branches. And cytokinins stop apical dominance. Usually, continuous growth of shoot apex, that is called apical dominance. So, in the apical dominance, stem grows very lengthy, but the stem won't produce lateral branches. So, that apical dominance activity takes place by auxin hormone. But here, cytokinins stop the apical dominance, means it stop the apical bud activity and encourage the axillary bud activity. That is why these cytokinins produce so many lateral branches and plants become bushy. Cytokinins stop apical dominance, and cytokinins promote nutrient mobilization, which leads to the delay of leaf flower senescence. Here at home, when you cut leafy vegetables, flowers, what happens? Within few hours, they will fade. But when you go to vegetable market, the leafy vegetables are very fresh for longer time. And when you see the flower bouquet and when you see the flower decoration in the functions, the flowers are very fresh for longer time. How it is possible? Because by spraying cytokinins, the leaves and flowers become very fresh for longer time, because these cytokinins promote continuous nutrient supply in the harvested leaves and harvested flowers. So what happens? This continuous nutrient supply leads to the delay of leaf and flower senescence. Senescence means, delay means here stopping, fading. In the next class, I will continue this lesson with the physiological effects of abscisic acid and ethylene. The 2 marks and 4 marks question and answers of this lesson in English medium are in the description. If you like this video, please share and subscribe to my channel. Thank you.